Hey everyone, and welcome to The Explainer. I am absolutely thrilled to have you here today. We are going to dive into a truly fascinating topic, the incredible ways our very own cells adapt to survive microscopic stress. I mean, just think about your own body for a second. You are constantly exposed to changing environments, varying workloads, unpredictable external pressures, right? But our cells? They don't just sit there and take it. They're highly dynamic. They literally have a built-in survival toolkit that allows them to shift, shrink, grow, or even completely change their identity just to stay alive. And today, we're going to break down exactly how they pull off these microscopic miracles.
Okay, let's just quickly look at our agenda for today. We'll start with surviving cellular stress, then cover atrophy, hypertrophy, hyperplasia, and metaplasia, before wrapping it all up with a final high-yield summary.
All right, let's kick things off with Section 1, Surviving Cellular Stress, the Need to Adapt.
So, the fundamental problem every single cell faces is that its environment is, well, never perfectly stable. Whether it's a lack of nutrients, a sudden surge of hormones, a physical injury, or just a changing mechanical workload, cells are constantly bombarded by environmental shifts. And to maintain homeostasis, basically to keep the peace, they absolutely need a survival strategy. So I really want you to think about this fundamental question. What actually happens when a cell is placed under stress? Does it shrink away to conserve its resources? Does it bulk up and grow to handle the heavy lifting? Does it multiply rapidly to share the burden? Or can it actually do something as wild as changing its entire cellular identity? Well, incredibly, the answer is all of the above, and it completely depends on the specific stressor and the specific cell type involved.
And that brings us to our core definition here. Cellular adaptation is a reversible change in cell size, number, phenotype, metabolic activity, or function that allows a cell to survive altered conditions. Now, I really want to emphasize that one word, reversible. It's an absolutely crucial recurring motif in pathology. Adaptation is a survival tactic. That means when the stressful condition is finally removed, the cell can often just return right back toward its normal baseline state. It's a temporary, but honestly, brilliant adjustment.
Moving right along to Section 2. Atrophy, shrinking to survive.
So, when cells face a reduced workload, a restricted blood supply, which we call ischemia, or just poor nutrition, they shrink. It makes sense, right? If the body can't afford to maintain a large cell, the cell downsizes. But it's not just some passive deflation. It's a highly regulated, very active process. To actually get smaller, the cell first decreases its protein synthesis. It just stops building new structural components. At the exact same time, it actively increases protein degradation. It literally tags its own proteins using something called the ubitin proteasome pathway, breaking them down for spare parts. On top of that, the cell uses autophagy, which literally translates to self-eating, to remove damaged organelles and cytoplasmic components. By systematically breaking down its own structure, the cell drastically lowers its metabolic demands and conserves precious energy.
Let's ground this biology with a few real-world clinical examples. This is essentially the classic, if you don't use it, you lose it principle, playing out right at the tissue level. Just imagine an immobilized limb stuck in a plaster cast. That lack of use directly causes skeletal muscle disuse atrophy. Or consider denervation. When a muscle actually loses its nerve supply, it rapidly shrinks. We also see this happen systemically, like in normal aging or with neurodegenerative diseases. Those can cause cerebral atrophy, where the actual physical tissue of the brain visibly shrinks down.
All right, Section 3, Hypertrophy, growing under pressure.
Now, what if the stress isn't a lack of resources but an overwhelming demand? Well, instead of shrinking, the cell has to bulk up. Hypertrophy kicks off when a cell experiences an increased mechanical workload or gets hit with specific growth factors. This stress activates mechanosensors on the cell, which then triggers some really complex intracellular signaling pathways. A major one you'll want to remember, especially for cardiac muscle, is the PI3K-AKT pathway, alongside a few other stress-responsive signals. Basically, these pathways command the cell to drastically increase the synthesis of contractile proteins and other internal components. The end result? The actual physical enlargement of individual cells.
To see this adaptation in action, we can look at both pathological and physiological examples. On the pathological side, think of a struggling heart. A patient with chronic hypertension is forcing their heart to pump against incredibly high resistance, constantly. So the left ventricle adapts through left ventricular hypertrophy. The muscle wall becomes massively abnormally thickened. But on the flip side, we have a healthy physiological example, pumping iron. Exercise training places a mechanical workload right on your skeletal muscles, producing a targeted, pretty spectacular enlargement of individual muscle fibers.
Section 4, Hyperplasia, multiplying for demand.
So, while hypertrophy was all about bulking up, hyperplasia is about multiplying for demand. But there is a really critical caveat here. This survival tactic relies entirely on a tissue's ability to actually enter the cell cycle and divide. If a cell cannot divide, it simply cannot undergo hyperplasia. And this brilliantly illustrates the difference between these two frequently confused terms. Hypertrophy is an increase in cell size, hyperplasia is an increase in cell number. Now, it's totally possible for a tissue to undergo both at the exact same time if the cells are capable of division, but fundamentally, these are two entirely different mechanical responses to stress.
The mechanism of hyperplasia works a lot like a powerful chain reaction. It usually starts when a growth factor or a hormone binds to a receptor on the cell surface. This activates the receptor, sending a wave of intracellular signaling deep into the cell. Think of this signal as a green light for cell cycle progression. The cell replicates its DNA, divides, and results in the proliferation of multiple daughter cells. You are quite literally building a bigger workforce to handle the incoming stress.
Here are your high-yield examples for hyperplasia. A perfectly normal physiological example is hormonal hyperplasia. Think about the proliferation of breast glandular tissue during pregnancy to prepare for lactation. Another truly amazing physiological example is compensatory hyperplasia. If you surgically remove a portion of the liver, the remaining tissue will rapidly regenerate through hyperplasia. But it can also be pathological. A super common example is benign prostatic hyperplasia, or BPH, where hormonal signaling drives an abnormal, excessive proliferation of both stromal and glandular cells in the prostate.
Next up, Section 5, Metaplasia, changing cell identity.
Okay, so we've seen shrinking, growing, and multiplying. Now we transition to what is probably the most dramatic adaptation, handling persistent irritation, but changing identity altogether. Now the crucial point is this. Mature, fully differentiated cells do not just magically transform into a different cell type. That is a very common misconception. Instead, when a tissue is subjected to chronic stress or irritation, the entire signaling environment changes. This altered signaling affects the tissue stem cells, or undifferentiated precursors. It triggers stem cell reprogramming, forcing them to differentiate along an entirely different pathway. The result is a brand new cell type that is much better equipped to tolerate that specific, persistent stress.
Let's anchor this with two incredibly classic examples you're going to see time and time again. The first is the smoker's airway. The delicate, pseudostratified ciliated columnar epithelium of the respiratory tract just can't handle harsh, hot smoke. So the stem cells reprogram to create tough, stratified squamous epithelium. We call that squamous metaplasia. The second example is GERD, or chronic acid reflux. The normal squamous lining of the distal esophagus gets repeatedly burned by stomach acid, so it adapts by actually changing into intestinal-type columnar epithelium, which is a condition known as Barrett's esophagus. But I gotta give you a vital warning here. While metaplasia is adaptive and reversible, persistent injury greatly increases the risk of dysplasia and ultimately cancer. This adaptation definitely has its limits.
And that brings us to Section 6, the final high-yield summary.
We have covered a tremendous amount of ground today. Let's rhythmically recap our rule of 4 to really lock down that framework for you. First, atrophy. The cell gets smaller because protein synthesis falls and degradation increases. Think disuse muscle atrophy. Second, hypertrophy. The cell gets bigger because stress signaling ramps up protein synthesis, like left ventricular hypertrophy and chronic hypertension. Third, hyperplasia. Cell numbers increase through growth factor-driven proliferation, seen in BPH or pregnancy breast changes. And finally, metaplasia. Cell identity changes entirely through stem cell reprogramming, like what happens in a smoker's airway or Barrett's esophagus. The core concept tying all of this together is simple. Adaptation means the cell fundamentally changes itself to survive stress.
But I want to leave you with a final, kind of provocative thought today. We know all 4 of these incredible adaptations are reversible survival tactics designed to keep the cell viable. But what happens to the cell if the stress becomes excessive, relentless, and simply impossible to adapt to anymore? When the limits of adaptation are breached, the cell crosses a very dangerous threshold, right into the territory of irreversible cell injury. And that fascinating destructive process is a story for our next explainer. Keep learning, stay curious, and I'll see you next time.